So welcome to the third short video that I've prepared for you about the factors that impact enzyme activity. This is part 3. Again, the references that I've given you are the same. The salad and text, especially that figure on page 980, was really good. The WISE Lab manual was not so good, but the introduction was not too bad. And on OpenStax, I've put a link for you here. Okay, so let's go ahead and let's get started. The need to knows here are really the purpose of these activities that we're going to be doing are to look at the roles of temperature, particle size, and pH on digestive enzyme activity. And really, the purpose of this short video is to orient you to the wet lab activities so that you can hit the ground running when you do get there. All right, so let's get started. The first experiment that you're going to be doing is designed to look at particle size and digestion rate. We're not going to be using an enzyme. We're actually going to have an Alka-Seltzer tablet stand in for our substrate here. So you'll have one Alka-Seltzer tablet here. They're round. And what you're going to do is you're just going to break it in half, and one half of that Alka-Seltzer tablet you're going to crush into a powder, and the other half you're going to leave in that kind of half moon shape. What you're going to do then is you're going to have 2 test tubes or 2 beakers, depending on how the lab is set up. You're going to put the intact half tablet in one beaker or test tube and the crushed powder into the second test tube or beaker. Now, if you haven't put your water in first, then you can go ahead and get those things transferred in there at the same time. If you do have water already here, you're going to want to add these tablets at separate times. The reason for this is that after you add the tablet, you're going to measure the amount of time that it takes for that tablet to dissolve. So in test tube or beaker 1, you're going to combine your intact half moon shaped tablet with water. You can stir it up with a wooden stir, or you can shake it gently. You're going to measure the time with a stopwatch or a timer on your phone maybe. When the tablet is completely dissolved, you can write that down in the activity handout that I've posted for you on ELC. Then you're going to do the same thing for the crushed half tablet. And you combine the powder with the water. You can shake it gently or swirl it gently or stir it with a wooden stir. And again, you're going to measure the time that it takes for all of the powder to dissolve. The second factors that we're going to be looking at, or the second group of experiments, are actually going to be looking at effective temperature and pH on enzyme activity. And we're going to start with this the first set of experiments here with carbohydrates, and amylase is the enzyme that acts on this. And here in this table, what I've given you is the substrate and the enzyme over here, and then the product over here. So when carbohydrates are acted on by enzymes, we see that the products are going to be monosaccharides or simple sugars or smaller sugar molecules like disaccharides. When we have a protein that's acted on by an enzyme, the products there are going to be amino acids or smaller peptides. And when we have a lipid that's acted on by an enzyme, and specifically here when we say lipid, what we really mean is triglyceride, we're going to be seeing fatty acids or smaller fat molecules like mono and diglycerides. So to test if an enzyme was actually working on our substrate, we could do a couple of things. The first thing that we could do is we could actually test to see how much of the substrate had been utilized. In this case, we would be measuring, say, the rate of disappearance of the carbohydrate. The other thing that we could do is we could measure the rate or measure how much of the product is being formed, how much sugar is being formed when we incubate our enzyme and our carbohydrate together. In the case of our amylase digestion of carbohydrates, we're actually going to do both. So we're going to be looking after incubation of our starch with an enzyme, we're going to be looking for starch in the test tube with something called the Lugol's test. And I think this will look pretty familiar to you after you see it. Additionally, we're going to run a second test, and we're going to be looking for the presence of small sugar molecules with something called a Benedict's test. So let's see what both of those are. Okay, our iodine or our Lugol's test is shown over here. You may be familiar with this in grade school when you were asked to put a few drops of iodine on a potato and you saw that it turned dark blue. When we add the iodine solution to our test tube here, if there's any starch that's present, you'll see this very characteristic inky blue-black solution that will be formed. If there is no starch present, then what you will see is that the solution will take on the color of the iodine, so it'll be this kind of reddish, goldish brown color here. The Benedict's test is a bit different. So the Benedict's reagent is this very pretty blue color, and it gets its blue color from copper ions that are present in the solution. The copper ions are a heavy metal, so you'll want to be careful that you don't dispose of your sample down the drain or down the sink. We'll have special waste containers for you. When you add the Benedict's reagent to your test tube and you heat it, you should see a color change if there are sugars that are present. And the color change can be something from kind of a yellow greenish color to this pretty orange color to a deep brick red color. That color change indicates the presence of sugars in your test tube. Now, in this image, this author is actually showing that they are heating the test tube with a Bunsen burner. We are going to heat your tube in a water bath because it's a little bit smarter when you've got as many students as we have, but the reaction will still go forward just the same. All right, so now your test tube setup for your experiment might look something like this. So what I've drawn for you here, I want you to imagine that this is a test tube rack and you've labeled 5 test tubes, 1, 2, 3, 4, 5 here. And then up here at the very top, I've put the incubation temperature. And then here in the second row here, what I've put are the constituents, what's in each tube. So in test tube number 1, we have amylase, which is our enzyme only. There's no substrate. In test tube number 2, we have no enzyme. We have starch, which is our substrate. Only starch is present. In test tube number 3, we have starch, which is our substrate, and we have boiled amylase, which is actually denatured. In test tube number 4, we have starch and amylase here. And in test tube number 5, we also have starch and amylase. The difference between test tube number 5 and test tube number 4 happens to be the incubation temperature. So right away, you should be able to see that we can test for the impact of incubation temperature on the activity of our enzyme here. We're also going to be looking for the ability of the reaction to occur in the absence of an enzyme. That would be what this test tube is testing for. And there's probably another factor here. If you were to look carefully, you might see it. So I want to leave you with a question here. In which test tube do you expect to see the greatest digestion of starch, and why? And what was that going to look like in terms of your Lugol's test and your Benedict's test? So take a minute and think about that. In which of these tubes, number 1 through 5, would you expect to see the greatest digestion of starch? And what would that look like in terms of the results of your Lugol's test and your Benedict's test? Okay. Now, the next chemical reaction that we'll be talking about will be related to proteins. Remember, when proteins are acted on by an enzyme, we just said they produce amino acids or smaller peptides. Unlike the first experiment here, if you're doing this experiment in lab, we're actually just going to do one test, and we're going to be looking for the presence of proteins here. And specifically, we're going to be looking for the presence of peptide bonds here, and we're going to be using something called a Biuret test. The Biuret solution is actually this kind of light blue color. When you add it to a solution where you have proteins, which will have a lot of peptide bonds, the solution should turn more of a violet purplish color here. In solutions that have relatively fewer peptide bonds or have no peptide bonds, you would expect to see this blue color, the same color as the reagent itself. Okay, so your setup for your tubes here will actually be pretty similar. So we're going to label 6 test tubes this time, number 1 through 6. In test tube number 1, you're going to have enzyme and water, but no substrate. In test tube number 2, you're going to have albumin, which is our substrate, but no enzyme. In test tube number 3, you're going to have albumin and then boiled pepsin or denatured pepsin. In test tube number 4, you're going to have albumin and pepsin and water. In test tube number 5, you're going to have albumin and pepsin and water. And in test tube number 6, you're going to have albumin and pepsin and hydrochloric acid. So notice here that we are also going to be studying the impact. We're going to be looking at the impact of temperature because we do have temperature that is varying. But we're also going to be looking for the effect of pH, as we've got one test tube here where the pH will be lower than the other tubes. And the question that I want to leave you with here is, in which tube, number 1 through number 6, do you expect to see the greatest digestion of albumin or protein? And what is that going to look like in terms of your Biuret test? So take a minute and think about that. Now the next possible test that you might be doing in lab is going to involve looking at the breakdown of lipids with an enzyme. And here we're going to be looking at the production of fatty acids, and we're going to be using something called a litmus milk test, which is a little bit different than some of these other tests, but it is exactly what it sounds like. So in a litmus milk test, what you're doing is you're actually incubating your substrate and your enzyme with some milk or cream and some enzymes. In this case, the enzyme we're going to be using is pancreatin. As the fatty acids are released from the lipid, then the pH of the solution goes down and the litmus powder that's present in your litmus milk turns pink. If you had a solution that was alkaline, then the litmus powder would turn more of a blue and you'd see more of a purplish color. So you're actually going to be looking for a color change. It looks pretty similar to what you would see in litmus paper. Your tube setup is going to look something like this. So again, we'll have 5 tubes labeled 1 through 5. In test tube number 1, we're going to have pancreatin, which is kind of a catch-all name for a collection of enzymes that come from the pancreas. So it will have both lipases and proteases that are present in here. In test tube number 2, you're going to have milk or cream. You can use either one. Obviously, cream will have more fat. In test tube number 3, you're going to have milk or cream and then boiled pancreatin solution. In test tube number 4, you're going to have milk or cream and pancreatin. And in test tube number 5, you're going to have your milk or cream and then pancreatin. And the difference here, again, is going to be the incubation temperature. And so let me ask this question now. In which tube do you expect to see the greatest digestion of fat? And what does that look like in terms of your litmus milk test? So take a minute and be thinking about that. Okay, this brings me to the end of my short video. What you should do now is take the pre-lab quiz. Good luck, and we'll see you in lab.